This video is brought to you by Vita Bee Health. Thanks Vita for sponsoring this channel. Please check them out, link in the description of this video. Hello everyone, today I have the pleasure of speaking with Dr. James Bartlett, a leading entomologist and evolutionary biologist at the University of Georgia. In today's episode, we discuss his career, how he became interested in honeybees, and his unique relationship with the beekeeping community in Georgia. Dr. Bartlett is beloved by this community and has their full support, which allows him to undertake honeybee projects that other researchers might shy away from or be reluctant to publish for fear of backlash from the pesticide industry. This is precisely the case in today's episode, where we delve into his latest publication, which for the first time, Dr. Bartlett has demonstrated that neonicotinoids actually help Varroa mites in killing honeybees in field trials conditions. This is a crucial conversation and I urge you to share it with all beekeepers out there. Many beekeepers still blame themselves for the high mite levels they have, even when they have done everything they can possibly do. And the message here is clear. It is not your fault. It is the pesticide industry, as I've repeatedly covered in this channel. And now I introduce to you Dr. James Bartlett. Lewis, welcome to the show, my friend. Long time no see. How are you?
I'm good. I'm glad to be here. It's been a while since we've connected, so I'm looking forward to talking about some bees.
Yes. Last time we connected, I think was in Georgia, right? The state meeting that you give a fantastic presentation.
Possibly, yeah. I mean, I speak at the Georgia meeting every year, typically. It might have been a few years ago now, though, that we were both there at the same time. Maybe it was just last year, though.
I think it was last year.
Time moves oddly at the moment. They feel like they're very long years, especially at the end of the beekeeping season.
And the older I get, apparently it looks like the time passed faster. I don't know if there's something with age, but yeah. Anyway, so since the last time we met, I want to talk about that first, and because I felt that you were so loved in Georgia in the state meeting, and that's not common for researchers at all. And I want to ask you, can you tell us a little bit of your background and your journey to get where you are, and how you you captivated so many people and people love you so much with
Yeah, so I'm very blessed that the Georgia Beekeepers Association especially is very supportive of me and the lab more widely. So my first interactions with them were really right at the start of my PhD. So I trained in England, where I'm from originally. That's where I first learned beekeeping 12 or more years ago now. And then when I started my doctoral research, part of that was always arranged that I would come out to the University of Georgia Bee Lab back when it was headed by Keith Delaplane and Jennifer Berry and others. And I'd do 2-3 months field work out here as part of this much bigger transatlantic sponsored research project that my supervisor was also on. So all the way back in 2014, right around this time of year, actually, it was just around Halloween, was my first trip to Georgia, and I started doing work at the Bee Lab just during the summers. So I'd fly from Britain to Georgia to do beekeeping, which was quite the temperature shock. And then halfway through doing my doctoral degree, my lab and my advisor moved to California, to UC Berkeley. So I actually moved and relocated. I still did all my field work here, but I was, for the majority of my work, was actually based at University of California in Berkeley. But it was still awarded and funded by my British institution. So I kind of had these 3 major universities that were all involved in my doctoral work. As we were coming towards the end of that, there was some money left on the grant, and Jaap de Rood, who is a professor at Emory University in Atlanta, asked if I'd postdoc on that grant for the remaining 9 months just to finish off some of the work we were doing. So that's when I relocated to Georgia, was 2018. And from there, kind of continued. But really, my connection with the beekeepers here started in 2015 with our first Young Harris Beekeeping Institute that I got to attend. So that was a 30-year span of Keith Delaplane and Paul Arnold and Jennifer Berry and a couple of others really building this bee school where they'd bring speakers in, not just from across the US, but they'd fly them in from everywhere across the earth to make sure that beekeepers were getting to speak to experts. It's where we started our master beekeeping programming. And at its height, we had 400, 500 people attending that institute. We'd have up to 8 or 10 parallel sessions at once. There were tracks for honey show training. There were tracks for certified, for journeyman, for master, for special interests. And it was a long meeting. We started typically on Wednesday and ran through to Saturday. And that was really my first exposure to the beekeepers here. It's where I started practicing giving talks and speaking about my research. And I was a young man back then still. I think I was only 23 the first time I presented at that institute. And I think that's part of why I've been able to forge what I view, at least, is a really good relationship with the Beekeepers Association here, because they've known me since I was quite young in my career. And that's allowed me to grow in a direction that they've helped shape me towards. And also I came in knowing very little. I was very upfront that I'd only been keeping bees maybe 3 years, 4 years by that point. And so I still had a lot to learn. And I was still early on in my doctoral work. I then got a fellowship at the University of Georgia to stay at the bee lab to test some of the ideas I had that had come out of my doctoral work. I was awarded that. That was with the Center for the Ecology of Infectious Diseases in our School of Ecology, which I still maintain a position with. And then as that 3-year postdoctoral fellowship came to an end, it was actually the Georgia Beekeepers Association, as well as the work of some of the people at the university who gave the university a call and said, we really don't want him to move out of state. Can you create a faculty position for him? Which they did. And Georgia Beekeepers co-sponsored the creation of that position originally. I've since been able to solidify it into a tenure track position, whereby they're not having to front any of the salary anymore. But the whole reason I was able to stay here in Georgia was that body of beekeepers essentially told me I wasn't allowed to leave. They sat me down and these lovely, very well-educated, very knowledgeable women of North Atlanta who've run a lot of Georgia beekeeping essentially said to me, you'll leave when we say you can leave, sweetheart. And I said, yes, ma'am. And they have installed me since, with Keith Delaplane's retirement, as the current, at least, co-director of the UJB lab. So without that very close relationship with the Georgia beekeepers, I doubt I'd even be a professor here in Georgia. I'd have probably ended up in a different state, honestly.
Wow, what a story. Well, I think that's part of what actually America is. When you create value here, things happen when you create value. And I think you are a researcher that creates a lot of value to beekeepers, and beekeepers respond. And beekeepers respond when you really create value for them. And I think your case is one of those beautiful cases out there. And congratulations, man. That was
Thank you.
Great.
Yeah, it's been a lot of fun so far, kind of inheriting the lab, changing a little bit of how things are done, and then keeping the things that worked really well the same. We now partner with Georgia Beekeepers on running all our master beekeeping programming. I give the same plenary every year, which is just titled Updates from Your Bee Lab, where I cover over all the students that have joined, that have left, what science we've published in the last year, what grants we've applied for, what grants we've won, what grants we haven't, to really make sure, hopefully, they have a sense of openness and transparency with how the bee labs run, because the beekeepers do donate a not insubstantial amount of money to the research program when we need it. And so I'd like to ensure that they as a body understand the inner workings of the lab, really. I think that's important for maintaining that relationship into the future.
I'm so glad you stayed in producing amazing quality work. And one of those studies that you were producing, I want to discuss here today because got my attention a lot. It's something, a lot of the concepts and the fundamentals of the study really reach what my audience and I talk about in this channel for a while, about pesticides and things that very few people like to cover about those subjects. And you published it just in this year.
Yes.
In the Journal of Insect Science, a very interesting paper that I want you to help us to understand it better in a language that only you, with your charisma and your, your way of speaking, can translate this to beekeepers out there.
Mm-hmm.
So can you just guide us through the experiments, the importance of the article? I'm gonna put the article on the screen right now. Just give me 1 second.
Okay, yeah. Yeah, this was one of quite a few papers published by me and by others in these semi-annual honeybee health science issues of what we now do with Journal of Insect Science. I think some years ago they were being done with the journal Insects, published by MDPI, but we've moved over to an Entomological Society of America journal. And a lot of these publications in these special issues come out of the American Bee Research Conference that we host at ABF every year. So we run a scientific conference within the American Beekeeping Federation meeting, and these semi-annual special issues of these journals come out where a lot of us are able to put these applied apicultural papers with knowledge that hopefully they'll be picked up by people like you, which you have done. So this was really the brainchild of me, Jeff Williams at Auburn, Debbie Delaney up at Maryland, I believe, and then Keith Delaplane as well prior to his retirement. And this is one of those interesting cases, and I'm gonna say this up front, where the paper that we ended up with and the findings that were actually interesting were not at all what we set out to test. And that happens sometimes with these sorts of big experiments. Is what we were really looking at is not what ended up being the main takeaway. So the idea behind these studies that we did that really culminated in this paper was that for a long time, Keith Delaplane in particular, Dave Tarpey and others, Tom Seeley, really focused in on how the genetic diversity within the colony, which very closely corresponds to the number of males that queen has mated with, determines its overall health. And I was not trained in entomology. I wasn't trained in beekeeping. I was trained as an infectious disease biologist, as an epidemiologist, and as a parasitologist. And there's been, since about the turn of the millennium, since early 2000s, a real focus on how genetic diversity, genetic heterogeneity, helps reduce not only the frequency, but also the severity of disease and parasite outbreaks. And this was really well demonstrated in rice, for instance, by a big Chinese study a couple of decades ago. And in most instances that we look for it, you'll see this clear relationship between how genetically diverse a population is and how vulnerable it is to a big infectious disease sweeping through, or a parasite for that instance. And that's been really well shown in honeybees as well. There's paper after paper after paper showing that the more drones a queen mates with and the more different those drones are, the more robust her colony is going to be in the face of Nosema, Varroa, viruses, all these diseases and parasites that we're continually battling. And that's not what we set out to show here. Rather, we wanted to ask, is the same true for how they deal with pesticides? And the idea here was maybe you have a subset of bees that have a father that gave them the genetics. That means they're really good at detoxifying pesticides, for instance. Like they almost can function then like the colony's liver. And so we really wanted to see whether this diversity element, this mating element that plays into queen quality, is able to buffer a lot of the negative effects we observe when our bees are being exposed to these agricultural chemicals, these pesticides. And in this case, we were looking at a mix of two neonicotinoids, imidacloprid and clothianidin, I believe, which are common in the agricultural landscape. And I think what's important about this sort of work is when it comes to nutrition, when it comes to foul brood, Varroa, small hive beetles even, there are interventions that beekeepers can take. We can put pollen or protein patties into colonies. We can feed them sugar. We can go in with amitraz, thymol, antibiotics, terramycin, whatever you feel like, to head off some of these acute problems. And we know that nutrition is a major pillar. We know that pests and pathogens are a big pillar. But then that ecotoxicological aspect of pesticides is something that we know is a huge problem for bees, but beekeepers don't have much control over. There's very little we can do in our apiaries to protect our bees against the pesticides out in the nearby environment, because that's out of our control. That's not an in-hive process. And so this was an endeavor to see if that was something that we could kind of recommend as a line of defense against pesticide exposure. And so this was an experiment that was replicated in Auburn, in Alabama, by Auburn University with Jeff Williams' lab, up in the Northeast by Debbie Delaney and her lab, and then as well in Georgia. And these were queens that we essentially manipulated the mating structure of. I won't go into the details too heavily of how we did this, but there was a lot of super sister queens, and then they generated drones that we then used to inseminate certain other super sister queens, I believe. And Keith Delaplane loves an artificial insemination experiment. It's something that I started learning how to do a couple of years ago. It's really difficult. I admit that people that are really good at it, I have so much respect for, because while I can pull it off, those queens really last very long. And the idea here was we could manipulate not how much sperm material each queen got. Every queen got exactly the same amount of sperm. What mattered was how many drones had contributed to and were represented in that dose that she got. And so some queens received, plenty of genetic material, but it was only from one drone. And then some others received a mixture that only had 2 drones that contributed. And then some others 4, 8, 16, right up to 32, which starts getting to the more extreme end of what we'd ever observe a queen naturally mating with it. In some cases, they go a lot higher than that, but they're very uncommon to observe. And because we'd raised these drones from queens of also known mating, we knew exactly what parentage went into each of these queens' daughters. So we had really tight control over what genetics was in there. We also did some kind of de novo modeling to see what was... we did some de novo modeling to see when certain brood ages would take on certain tasks. And that's actually plugging into a separate paper that we did that was also published in this same journal. Another way we went about this was, because beekeepers can't necessarily do the AI themselves, is you can recreate that difference that we've done in other experiments that I just described by mixing brood or exchanging brood. And so if you have a population of bees in an apiary, and you know that those queens, for instance, are from distantly related sources, maybe you got some from one breeding program, say Saskatraz, maybe you've got some Russians, maybe you've got some South Georgia queens, maybe you've got your homegrown mutt queens, they're all in an apiary together. One way you can recreate that effect that I discussed of AIing the genetic diversity in the colony is simply to swap frames of capped brood. And when you do that swap, you temporarily increase the genetic diversity in the colony. And we were hoping that could be a lot more achievable for beekeepers. So we did a whole series of experiments as part of this FAR-funded project. And for this one, I'm going to focus in on when we did the brood swapping, but the results are pretty similar. And so we had some colonies that were just as normal. All the brood in there was from the queen that headed the colony. And then we had half the colony where we transplanted everything. And typically we were looking to mix between 3 and 6 frames. So we had apiaries that were either 12 colonies or 24 or whatever. And we'd move brood between them. At the same time, we put pollen traps on the front of all these colonies and then force-fed them pollen cakes that were spiked with known amounts of the pesticides that we were using. And this was pollen that had been tested as pesticide-free using, I believe, mass spectrometry in this case. And then we intentionally mixed either two neonicotinoids into it simultaneously, imidacloprid and clothianidin. And so then we end up with this 4x4 design, or 2x2 rather, this fourfold design here, where colonies are either genetically homogeneous and being fed safe pollen, which is the top left. They're genetically homogeneous and having to deal with this pesticide stressor because the only pollen available to them was what we were providing them, because we had pollen traps on the front of everything. And then we had the counterpart where we'd intentionally increased temporarily the genetic diversity of these colonies. And then we were also seeing whether or not they could cope better with this spiked pollen. The idea here was that the difference between colonies fed safe versus toxic pollen would be much bigger. So comparing across the top row than it would be in the lower row. That is to say, the effect of the pesticide should be smaller in the bottom row. And I'm just going to go ahead and say that is not what we found, unfortunately. We couldn't see any what we'd call interaction effect, whereby the degree of mixing influences the effect of that pesticide, which was a little disappointing because that is one fewer thing that beekeepers can do to protect against pesticides. And I think it highlights how difficult pesticide problems are to deal with in bees, because if something as fundamental as the genetic diversity of the colony doesn't seem to do much in reducing these negative effects, there's very little else that we can think would, beyond just making sure the bees are otherwise healthy, which we're all trying to do all the time. Now, in measuring the impact of this pesticide, we measured a bunch of different things about these colonies. We measured size, we measured aggression, we measured how much pollen they were bringing in, we measured larval survival. I think we had the queen survivorship, replacement. I think we had a list of 18 to maybe even 20 things we were measuring about all these colonies. And the one thing we found that the pesticides did, it didn't influence aggression. It didn't influence brood area. It didn't influence larval survival. It didn't influence how much pollen they were or were not bringing in. What the pesticide did do was make Varroa problems in those colonies worse, where the colonies that were given the pesticide pollen mixture had bigger Varroa populations measured by sticky screen, and they had higher Varroa parasitism rates. And those two are almost always correlated, but occasionally they can be different. If you've got a really massive colony, you might have more Varroa, but fewer Varroa per bee. Whereas in this case, it was both at the same time. The colonies tended to all be about the same size. And then the ones that had been fed this neonicotinoid mixture had more Varroa in them. And at the same time we were doing this experiment, there was some phenomenal work being done in Italy that I didn't know about that they published in 2020. This was by Anosia, DePrisco, and a few others, published in Nature Communications, where they were doing laboratory feeding with one of these same insecticides, one of these neonicotinoids, clothianidin in their case, and then they were intentionally letting Varroa feed on pupa or pre-pupal larvae that had either been fed brood food with pesticide in it or brood food without. And they did a lot of quite modern genetics, what we call transcriptomics, to look at how the bee is regulating certain genes. And they had found that the sublethal clothianidin dose that they were being fed was an immunosuppressant. It made their immune systems a lot weaker. And they showed that using these beautiful laboratory experiments, and they showed that those immunosuppressed bees were better for the Varroa. A Varroa foundress that feeds on a pupa that was fed one of these pesticides tends to have more daughters because the bee's immune system is less able to fight off that Varroa mite foundress and less able to poison the daughters that are also feeding there. And I think that provides a really clear mechanism to explain what we found, which was the field experiment counterpart of that, where the main effect of these neonicotinoids when they're being sublethally consumed, when they're not killing the bees outright, but they are making them unwell, is not that it causes larval death. It's not that it causes bees to be less efficient foragers. It's that it makes Varroa worse. And that's not something that it turns out had been shown experimentally in the field. We have loads of field evidence from studies that I'm on and plenty that I'm not, linking pesticide exposure to Varroa. This was one of the first times it had been intentionally manipulated in the field. And we had this phenomenal paper come out by a completely separate group that provided an obvious mechanism as to why that would happen. And so while the genetics was what we really set out to study here, how colony genetics shapes the response of the colony to pesticide, what was actually the really interesting thing when we finished this experiment wasn't really anything to do with the queen or the genetics at all. It's that the main negative effect of these pesticides was that it made Varroa and disease worse, which I think is interesting for a bunch of reasons. One of them is beekeepers from the southern tip of Florida up to North Dakota all routinely report the same thing, and that's that they can treat two apiaries exactly the same, and one will be doing absolutely fine, and one is just being hammered by Varroa or viruses. And I see this in our apiaries as well. We can keep exactly the same bees in exactly the same way, but one of your apiaries consistently has a Varroa problem when another one doesn't. And I think findings like this begin to explain that it can just be a difference in, a subtle difference in environment. You're not seeing any pesticide kills. You're not seeing bees that are trembling. They're not dying with their tongues out. All it requires is a small amount of this pesticide closer to one apiary than another, and you're going to see the effect of that as worse Varroa. And so I think it helps explain some of the things that a lot of beekeepers find frustrating, which is why is one apiary or one set of bees doing so much worse than another? I think it is these very subtle agrochemical effects that are really only then being noticed in the form of an alcohol wash. I do think it also has a positive message in an underlying element of what I've just discussed is how hard it is to protect your bees against pesticides. But if the main effect of pesticides is levied via parasites and diseases, then we can actually reduce the strain of those pesticides on the bees by making sure that our Varroa are absolutely controlled, that our foul brood is under control, and that all the things we do to protect them against these parasites is also indirectly making them much more resilient to pesticides, because the neonicotinoids can't increase the Varroa number if there's almost no Varroa in there. And I think that gives us an element of hopefulness that even if we can't stop the bees bringing the pesticide into the colony, bringing the toxin to their larvae, if the only thing that toxin does is make Varroa worse, it doesn't matter that it's there if you have almost no Varroa. And so that's some of the beekeeper element I've brought out of this is, yes, it's frustrating that we can't improve the colony's detoxification, that we can't stop them going and visiting flowers that have these toxic chemicals on there, but we can take care of their other problems, and that makes the pesticide less important. And so that's really what I've taken from this. And we should have we've got a couple of graphs kind of showing the difference in Varroa levels between these treatments. And this was a short experiment. I think it was
Please guide me here.
8 or 12 weeks. And so this occurred quite quickly. And so we saw just from that mess of statistics there, this was this wasn't anything to do with Varroa. This was kind of proof that our genetic part worked. So one of the really, really common findings when you have colonies that have greater genetic diversity, more patrolines represented amongst their workers, is that larvae tend to survive at higher rates. And we do that by going in and marking where there's an egg or marking where there's a first instar larva on a clear sheet of acetate that goes up against the frame. And then we come back 2 weeks later or 10 days later, really, and we see how many of them aren't capped. And if they're not capped, you can discern that one of those larvae died at some point. It's likely been replaced, but if it's not capped, it wasn't, it didn't survive through. And a really consistent result is that the more diverse your nurse bees, the more likely larvae are to survive to pupation, to survive to their cell being capped over. So here, what we really want to do with these graphs is, in this case, is compare the pink on the far left to the blue, and then also compare the red to the purple, because that's the difference as to whether or not there's this genetic mixing going in. And what we see there is that the mixed colonies, so comparing pink to blue or comparing red to purple, tend to have higher larval survival. And that was something that has been found by loads of other papers. And so it was just a useful confirmation that what we were looking for happened. So the experiment worked in that it found a result we were expecting. Now, if we go further down to the Varroa results, this is really where we start talking about the negative effect of the pesticide. And you can see the colors have been rearranged slightly to make those comparisons that I talked about easier. So now what we're actually comparing is pink to red and blue to purple, because that denotes the addition of the pesticide. And what we see in both cases, so on the left, that's the sticky screen data, and on the right, it's alcohol washers. And these aren't actual numbers on these axes. These aren't the numbers of Varroa on the sticky screens. Obviously, we're not getting -1 Varroa on the sticky screen.
Yep.
These are standardized to the average colony for that apiarian site. And that's because we had, as I said, this experiment running up in Delaware, which is where Debbie Delaney is. I think I said Maryland earlier. Apologies, I'm not American. I still can't tell a couple of my states apart. Up in Delaware. And then we had replicates down in Georgia, and we had replicates over in Alabama. And within each of those places, we also had different apiaries. And so in order to display all that data cleanly at once, we have to do some funny stuff with the y-axis. So the numbers that you'll see on the y-axis here aren't real. That's just a measure of the effect size. But what we see consistently with both the sticky screen work on the left and the alcohol wash on the right, and it's clearer on the alcohol wash, is that the addition of the pesticide, so moving from pink to red or blue to purple, increases the number of Varroa we're finding. And that was the crucial, useful bit of this study that we didn't even realize we were the first people to test. Honestly, I was shocked that there hadn't been field tests of neonicotinoids sublethally on Varroa, because it's something that's been observed so many times through more observational work, through laboratory stuff, but this was the first field manipulation of it. And it just very clearly showed that the pesticide increases how many Varroa are in there over a very short amount of time as well. And as I said, there was nothing else that we found it did. We didn't find it changed colony aggression. We didn't find it reduced foraging success. We didn't even find that it reduced larval survivability. All it did was make the Varroa problem worse. And so that was the main takeaway from this paper, and something that has been a new part of what I work on. Yeah.
I need to stop you, because you mentioned you were shocked. I'm gonna tell you, you don't need to be shocked. I'm gonna tell you, you are the first one with the guts to publish. I know other ones that was not published.
I did suspect that other people must have looked at this.
They have done,
and because neonicotinoids are a hot topic. It's a difficult arena to step into. These are very charged topics. And if you come out the door and say, hey, this pesticide's maybe not as bad as we thought, because that's the other big measure here is it's not doing too much. I had a paper out also last year that looked at the effect of neonicotinoids on monarch butterflies, led by Cody Prouty, who's now actually at the Bee Lab down at University of Florida. He did his master's here at University of Georgia. And similarly, we found that this pesticide wasn't that bad for the monarchs. They really didn't care about it. And those papers tend to be more nervous about publishing them, I think, because we don't want to say something's safe if it's not. But I also think we do need to honestly report to our beekeepers especially when something isn't maybe as bad as we thought, when we don't have to worry about something as much, even if it's maybe not the messaging that we expected to see.
Fair enough. And I think that's exactly why I think the beekeepers are investing on you in Georgia. You're creating the value and you have the guts to publish what other people have done in the past, and they did it. Mm-hmm. So when I saw this published, I said, who did it? I'm very proud actually, because I have huge fights in the backstage of other places. Let's not put names on it.
No.
And yeah, so that's why I like the way the people support you, so you can contribute with the things that you see clearly, with transparency, with no interference that is all over the place. But I wanna pick up your brain now a little bit, because now there was a clear, very well designed experiment that very well controlled with the genetics, with something that normally we cannot do in the field very well. Very few people and facilities can have the capability to do this kind of results. And with only 2 neonicotinoids, when 2 pesticides, you could see some very impactful results. But the reality out there. There are papers from Dr. Pettis in the past showing hundreds of different pesticides interacting at the same time.
Absolutely.
What's your thoughts on that? Because all those combinations can create a diverse, in my opinion, of course, and everything end up in the lap of the beekeepers. The beekeepers are at the fault of everything. And I have to say, for you guys at home, if you're a beekeeper, it's not your fault. It is really hard to be a beekeeper today, because we have scenarios now with all those pesticides out there interacting with each other, creating all kinds of situations that is very easy to the people to slip away with their thoughts about things. And I think pesticides is a gigantic problem that we don't talk about. And I want to pick up your brain on that. Like, I see the same problem. I can see the same problem with any other, like, when you have those virals going up so badly, and we have absolutely no understanding why, and the beekeepers have done everything they can, in my opinion, that could be against one of these interactions of something. Could be a pesticide with a specific part of a plant, a pollen of a plant that can create a synergistic effect and can create problems. What's your thoughts on that, and how can we mitigate, if we can, other than maybe convince the people that we need to change the way we grow food? But let's see. I want to hear your thoughts on that.
Yeah, I know. I very much agree that often these outbreaks that we attribute to a specific disease are actually the roundabout effects of them getting into some specific contaminated feed that has this pesticide element. And we see that all the time at my lab. Because we do Varroa control work, we often have to hold colonies at quite a high Varroa level to then be able to go in and test new Varroa controls. And that's a real difficult knife edge to balance on, because we lost 2 whole apiaries this year because those bees were fine until they quickly died. And we're essentially certain it was that they all got into something that had just been sprayed on some cotton nearby. And it kind of highlights, again, that thing that you can get this sudden change in the stability of the colony just because you've got a little bit more stress. It's very a straw that broke the camel's back. And that's just in the terms of, having some low-level infectious disease burden that then becomes out of control when you have the pesticide on top of it as well. As for the pesticide mixes, there's people trying to work on that, but I think we're seeing time and time again how little we understand the parallel kind of effects of multiple pesticides. And a lot of my small hive beetle work at the moment is on chlorantraniliprole, which is a bee-safe insecticide, but it's not safe when it's co-applied with a fungicide. And that's work that Reed Johnson did up at Ohio State. It was what was driving a lot of colony loss in almond pollination. And in recent work as well that Frank Rinkevich led with Tom Rinder and a couple of others up in Iowa, I was brought on to do a lot of the statistics for. they were looking across a huge range of pesticides, and it wasn't just the insecticides that were important, it was the fungicides that were really obviously having an effect when we kind of put this massive amount of data looking at 40 or more different pesticides in the wax, in the honey, into this kind of statistical engine that churns out the results. And fungicides typically aren't toxic to bees. The testing that we have to do to get them labeled, the testing that we have to do to get them sold and EPA approved, it requires honeybee toxicology, but the fungicides on their own are rarely capable of harming the bees, but they have this very tractable, consistent influence on hive health when there are other pesticides, when there are other diseases in the environment. And I think that's really where the difficulty of this comes in, is it's often chemicals that aren't on their own at all harmful to bees, but they make the bees much more vulnerable to other stresses and to other pesticides particularly. I normally talk about this as being kind of like the grapefruit effect, where there's a lot of medications that if you're on as a person, you cannot drink grapefruit juice. Now, obviously, grapefruit juice is not toxic to people. Millions of people drink it every day and there's no harm. And the medications that we're taking are for our health. They're to regulate, whether it's your blood pressure, whether it's your cholesterol, who knows. But if you do both of those together, suddenly you're in trouble because there's this biochemical interaction mediated by your own body's either uptake or enzyme metabolism. And I think we're seeing that happen in bees all the time, but understanding the mechanistic basis for that, understanding when and where that crops up, I think is going to be a real new focus in bee health over the next 10 to 15 years, hopefully. I'd like to see it be really centered as a topic of keen interest, because, and I'm not the only one saying this, there's plenty of opinion pieces, there's plenty of good work out there from lots of people, both in Europe and the US, highlighting that synergistic stress as a problem. But right now, we kind of have to shrug and say, well, if it says it's bee safe on the bottle, you spoke only a moment ago, Umberto, about changing the way people farm and grow their food. We can't blame farmers, in my opinion, if that pesticide bottle has a giant thing on it from the EPA that says safe for bees. We can't blame them using that because they're following the label. They're doing what they're supposed to do. But it might be that, yes, that one's bee safe and that other one's bee safe, or yes, it's bee safe on its own, but not if you have, Nosema in the colony. How do we go about actually testing that and regulating it? And I think that's going to be the next big hurdle, because I agree. I think these cocktail effects, these multiple chemicals at once are underpinning a lot of the trouble we're seeing, but piecing those out is difficult. And I'm glad there's lots of other labs working on it, because I'm definitely not a toxicologist by training. I have to be a little bit to understand what's happening in the diseases, but when you really get into the nitty-gritty of what's happening physiologically, biochemically inside the bee, there's plenty of people who are better qualified to do that than I am, and I'm glad to see they are doing it.
It is a fascinating topic, and I am concerned about those things. When I think about this, I have nightmares sometimes. And the reason is simple. And you mentioned that I agree with you regarding the farmer putting something in their crop is a tool. Chemistry is a very powerful tool. And when you have these powerful tools, we need to regulate it better, I would say. But do you think it will be should we blame the farmer if his stew end up in the water that we drink, for example? We have data showing that most of those toxic chemicals end up in waters and rivers and places like that. And that interacted, we ingest, our kids ingest, my dog ingest, there are clinical trials about other drugs, trying to cure cancer. And then maybe these chemicals that you're not supposed to be there, interact with stuff and messed up the clinical trials. There is a big mess when these powerful chemistry tools are not restricted only in a spot they're supposed to be acting to. If the farmers are growing food and they're able to control the spread, and you can guarantee it doesn't go anywhere, and then the bees go there and get killed, okay, no problem. That's, the bees invade somebody else's area. I'm fine with that. The problem is when you have your dog and the dog jump the fence and start to, bite people, then people got sued, but not in this case. So thoughts on that?
Yeah, so I have a very strange relationship with regulation in some ways, because from what you just described, I agree. I think we need firmer and more effective guidelines for how to prevent leaching and ubiquity of these chemicals, because if we're particularly looking at the impacts of their synergy of the mixtures, if you can't even keep the chemical on the farm, then we're already in a lost cause, because you can't know what the bees are getting in their water source or from wild plants that haven't even been spread. On the other hand, we know how difficult it is to get new treatments for bees approved. And it's a challenging argument when we're asking for more regulation of pesticides on one hand, when it comes to crops and the environment to protect our bees, but then we're also asking for easier regulatory environments to be able to get new varroicides, new small hive beetle controls, new antibiotics into bees. And that's a hard discussion to have when we're asking for two conflicting things, right? More regulation and less simultaneously. I think it's very easy for other parts of the agricultural industry to then look at us and think we're asking to have our cake and eat it too. And so I don't think there's the simplest solution there, but I do think it needs to come from the regulators, and I think a lot of it needs to be at the state level, because it is so crop to crop different and so environment to environment different. You go to Iowa, where they have some of the highest intensity corn and soy production in the country. They have an excess of water in those fields, which makes controlling leaching hard because they're having to drain those fields. That's very, very different to an environment where you're having to consistently irrigate. And so I think it can't be blanket federal rules. I really think it needs to be crop and location specific. As to the farmer's individual responsibility, I think we could do a better job of really educating about what the different classes of chemicals are and why that matters. And I hope I don't make any enemies saying this on your podcast, but I think we need to really do better at challenging the organic lobby, because the difference between a pyrethrin and a pyrethroid is essentially non-existent. One is just produced by us, and the other is produced in the plant it was isolated from. Neonicotinoids are called what they are because they're just new nicotines, right? They're nicotine molecules that we have tweaked slightly to either make them easier to produce or to reduce their impact on people or other things we're worried about. And I think we need to get more serious about talking about each individual chemical, each way of applying more seriously, and not just kind of wave our hands and say, oh, well, that one's organic, so it's fine, because it's often not. Plenty of toxic things exist in nature, and we often co-opt them. And I think until we can do better at drilling down into the individual things being applied so that farmers can make slightly more informed choices rather than just going off this play sheet of, oh, this one's organic and be safe so I can use it. I think we need to really give them the information necessary where they can make a more specific choice about, okay, well, here's how often I have to apply this one. Here's how water soluble it is. Here's how stable it is. And rather than having these big umbrella terms like be safe or organic, actually give them the information to really choose what is the right pesticide for that moment in time, for that environmental condition, for whether they're going to be irrigating soon or not. All these contexts. And I think we can trust farmers to do that. I don't think we've done a great job of giving them the information to be able to let them make that choice. I think there's an addiction to easy rules that we need to step away from in beekeeping as well as the rest of agriculture, is my current opinion.
Quite interesting. Thank you for sharing that. I want to pick a brain about another subject too that is always in my mind, and you are so well spoken, and I'm gonna take the opportunity here to ask you this. In the past, I remember Dad and many other old generation beekeepers. Normally, the way we solve a problem with the bees, we just let the bees solve the problem themselves with breeding. The bees, put a lot of bees in one area. A lot are gonna die. Take a time until we reach in a position that we have a genetic kit that is specific for the area. Basic genetic stuff. Yeah, and we know that worked for many times, and we have a lot of data on that. Even the beekeepers agree that that was the way they used to do it 50, 40 years ago.
Yeah.
Right now, this is not as easy as anymore. And normally I like to talk to beekeepers, and they ask me why. And I have a tendency to say that breeding in today's environment is nearly impossible, because you cannot, with the amount of pesticides attacking every single level of the bee's immune system or physiology, there is no way for you to breed for everything.
Yeah.
And I wonder your thoughts on that.
No, I agree. And the bee breeding stuff is something that hopefully, fingers crossed, I'll have a paper out soon with Nina Sokolov and my old lab at Berkeley, discussing that specifically within the context of viruses and Varroa. But you're right, it's ubiquitous. It's across all the stresses that they face in the landscape, whether that's periods of dearth like we see here in the southeast. I think we don't talk about that enough as a stressor, that there are months and months in the summer in the south of the US where there's nothing for them to eat. That, in addition to all the pesticides you mentioned, in addition to all the new problems like Varroa, like small hive beetles, and anything else that pops up, we're asking bees to be good at too many things simultaneously. And as someone who has done a lot of insect evolution experiments, that was a big part of my PhD, not in honeybees, but in a pest species of agriculture, looking at how much harder it is to evolve resistance to something if you are also nutritionally stressed, or if you are also coping with a toxin. And we use that all the time in resistance management, even in very simple cases. That's kind of the whole fundamental of IPM, is that you are rotating controls because your pest species find it much harder to evolve or adapt to lots of things at once. The exact same applies to species we want. We use that all the time to make life hard for pest species, but it's equally true for our bees. And so if we want them to be friendly, great brood pattern, great honey producers, not swarming, resistant to Varroa, resistant to viruses, immune to all these pesticides, of which there are many, that is a lot of things to ask one population to evolve towards. And I agree, I don't think that's fundamentally very achievable without really complicated and kind of lab-driven genetic screening, intentional big experiments, the kind of thing that, they started the Baton Rouge lab with the Russian bees, that the Saskatraz bees are famous for. But those are keen geneticists who started that. That's not a backyard operation. And I think what it means is that beekeepers just have to get realistic about what they're willing to have as their costs and what they want to prioritize as their benefit. For most of our hobbyist beekeepers, I don't think they care that much about how much honey they're getting off those colonies. They just want a little bit because it's fun. Most of our hobbyists, I think, fall off the hobby because it's really upsetting to see your bees dying as much as they do. And so I think you really need to prioritize survival there above most other things. That might mean you get some slightly defensive bees that aren't the most pleasant to keep. And then you have to ask yourself the question of, well, am I willing to pay the price of kind of spicy bees if it means that I can reliably have them abundant and they're not sick and they're not upset? And that's not going to be for everyone. You know, some people will make a very different decision. Where for one of our test apiaries at the moment, we inherited kind of 26 colonies that hadn't been even looked in since August last year. So it'd been a full year where they'd just been left in an apiary. The beekeeper had called up a friend and said, I'm too old for this now. Can you take them? I feel bad. I haven't looked at them in a year. We needed an extra apiary to do some Varroa testing. And so we kind of struck a deal where we moved the colonies for her, and then we got to use them for Varroa testing, Varroa control testing, Varoxan and Amaflex, which are both now on market. We're testing that at the moment because those should be full of Varroa, right? Because they hadn't been treated in a year. For a full half of those colonies, about 12 of them, we couldn't find a single Varroa mite, not on a sticky screen, not on an alcohol wash, not in brood. We even did the 200 cells uncapped. But I'll tell you now, those are some mean bees, and they're full of honey and they're surviving really well. You know, they're so populous, but you open that hive up and you are seeing 20 to 30 of them trying to sting you pretty immediately.
And you need to convince me that.
Yeah, it takes a lot for me to get flustered in an apiary now. You know, we run up to 300 colonies during the peak of the season at the bee lab. We're not a small lab. We're not like a commercial beekeeper, but we still know what it's like to have hundreds of colonies to get through. And I've been doing this a long time, like I said, you know, well over a decade now. And these are some of these bees are one of the few where I sometimes have to step away to kind of get them off me and let them calm down because they become so frenzied. Now, some of my beekeepers like the challenge of colonies like that. They find it fulfilling, I suppose, to have a little bit of adversity when beekeeping, particularly if it means they're getting loads of honey off those colonies and they're really resistant to everything in the environment. But a lot of my beekeepers would not put up with a colony that behaves like that. And I think that highlights this constant tension bees are under from an adaptionist point of view. Nothing comes for free in nature. That's one thing evolutionary biology teaches us over and over again. If you want to be good at one thing, probably going to have to pay a price somewhere else. And we know from studying feral bees that that's often swarming, it's requeening, it's brood breaks, it's aggression, or it's less honey production, or it's a slower start in spring. And I think we need to really just come to terms with the unfortunate reality that we're never going to have a perfect bee. There's going to be no silver bullet bee breeding program that provides the bee everyone wants. And the sooner we accept that, I think the easier it'll be to have conversations where people aren't trying to say, oh, well, I always keep this kind of bee because I find it's the best. What's your best? Where are you? What does your year look like? What are you trying to get out of these bees? And I think that's a hill that I am seeing beekeepers confront and get over. And there's such phenomenal writing from, you know, Tom Seeley and others about this that really starts them thinking along that process. But it's a difficult thing to confront because we all have our favorite hives, right? We all have our favorite colonies that we go out and we know that queen and she's your favorite and she's your best and she's killing it. That does not mean she'd be someone else's favorite queen. By all means, a bad queen, everyone hates, right? If a colony is dying, no one likes it.
Yep.
But accepting that what you consider to be a really good colony isn't universal is going to be the next step, I think, to really thinking about how we do that kind of breeding in the face of all these different stresses.
Wow. Well done. Yeah. A lot to unpack. I think that's why one of the things about beekeeping is the variety of things that come up in those conversations, because there's so much things going on at the same time, and that's, I think, is part of the fun of bees. Yeah. And you summarize things with such a is a art. I'm jealous of that kind of skills.
Well, if you ask any of my family, any of my friends, any of my colleagues, talking's one of my favorite things. I will just walk around my building looking for people to talk to. I've never been the sort of scientist that wants to be in the lab on their own for 10 hours. I've never been the sort of scientist who wants to be left alone. And so I suppose the benefit of that is that when I come on platforms like this, at least I know what to say, because I'll talk to anyone.
Very practiced a lot.
Yeah.
Lewis, one more thing before we wrap this up, what's coming from your studies now? What things are coming in the near future for us to pay attention to, and your final message to the people at home?
Yeah, absolutely. So I only just recently kind of inherited the bee lab. Depending on how you count it, I'm either in my second or third year as a professor, so everything is kind of at that exciting, new, but very stressful stage. The lab's pretty big. We've got anywhere between 6 and 8 graduate students, depending on how you count it, multiple staff. So there's lots going on. Jennifer Berry, who's in the lab with me, is doing a lot of great work on improving forage habitat for pollinators, not just by saying we should plant for pollinators, but by looking at instances where we're doing landscaping, things like stabilizing agricultural soil, where there are these specific seed mixes to do that job. How can we improve those to also provide food for pollinators? Because if a farmer's putting a bunch of seeds in the ground to secure the soil, let's see if we can find seeds that do both that and provide bee food. And I think that's the clever way to go about that. So I'm really excited to see what Jennifer, Dr. Berry, does with that work. We have a bunch of Varroa research going on. That's part of this big Honeybee Health Coalition-led Specialty Crop Research Initiative grant that has me and 7 other institutions, 9 other PIs on. We're testing, as I said, Viroxan, Amaflex, and a few other new products right now as part of that. Some of my students are really leaning into the effects from a disease perspective of what we feed our bees and when, looking at high fructose corn syrup compared to sugar syrup, compared to nothing at all, as well as how safe or dangerous it is to overfeed protein patties. And part of that is linked to a lot of my work right now, which is on controlling small hive beetles. Now, you've kept bees in the hot, humid, tropical southeast, Humberto, so you know how irritating the beetles are. A lot of my 5th, 6th year beekeepers here in Georgia say beetles are a bigger problem for them than Varroa. And we're seeing more and more the beetles start to become common, start to become a problem in kind of the southern end of what I'd call the Great Lakes states, southern Illinois, southern Indiana, western Kentucky, parts of Missouri. You're starting to see beekeepers encounter these beetles at levels they weren't traditionally used to. And so I'm excited to get a lot of the beetle control work moving along off the ground with respect to that as well. That's a quick flavor. There's plenty of other things going on in the lab as well, but those are the ones that I'm excited to see progressing. Is how does what we feed our bees and when matter for their diseases? How do we control small hive beetles effectively, safely, and legally? And what new Varroa stuff is on the horizon, and how do we get people to put more bee food in the ground without it being of extra cost to them? Those are the things I'm really excited to see mature over the next few years. And I have some other things that I'm playing around with, that I'm exploring, but they're in the earlier stages where you're still trying to figure out whether the science is even plausible. But we've got really good in the lab now at grafting and rearing larvae. We can take a first instar larvae and rear it in the lab right up to right through pupation to when they're actually emerging as adults. I'm sure you've done a lot of that in your time as well. And it's satisfying once you get good at that. It really opens up a whole new realm of experiments you can do once you've got grad students who can do that without it being a problem. So I'm excited to see what gets funded and what ideas my lab members bring to me as well. It's fun to see the next generation kind of coming in and flexing their ideas and flexing their muscles about what they think I haven't thought about before. So lots going on.
Oh, very important subject. Regarding small hive beetles, I was surprised. Maryland State Beekeeping Association asked me to give a talk about small hive beetle. So I give a little overview about what we know so far and the biology a bit. And I was there expecting to be an empty room. Boy, it was packed.
Yeah.
And I was just, what's going on? What's going on? People start to tell, we see small hive beetles everywhere now, so we need to know how to control. So, wow, I wasn't expecting that. So, so we need more research on small hive beetles. So thank you for taking that subject seriously.
Yeah, they're one of my earliest kind of research endeavors as a professor, and I think until I can get rid of them, I'm going to have to stay working on them, because while I find the adult beetles kind of fun to have in the lab in that entomologist way, they're just
it's easy to grow.
They're easy to work on, and the adults I think are quite charming. Unfortunately, I think they're kind of cute, but the larvae I do find still absolutely vile, and we've had a lot of colonies slimed out this season already, particularly weak colonies that are our control group for some of these Varroa studies. And so that's something that I'm keen to stay on. And my more senior graduate student, Ian Collins, he's the one leading that small hive beetle control work now for his PhD. And this year he started giving his first talks on that research to beekeepers as well. And so it's satisfying for me to see my lab members stepping up and learning how to communicate their science to the stakeholders as well.
That's great news. Looking forward to see all those results. So I'll make sure I'm going to report everything that's coming from your lab from now on. You're doing fantastic work. Any final message to the people at home before I wrap this up?
No, just thank you for keeping bees, and don't just rely on the internet for your information. Talk to your neighbors as well. What's a beekeeping problem for you is probably a beekeeping problem for the person 2 houses over. Might not be the same for someone 2,000 miles away. I think the fact that we have this sort of internet platform to get the science out there, to get people talking, is great. It's never a substitution for chatting to someone who's kept bees in your neighborhood for the last 15 years.
Close by, yeah. Similar environments, similar problems.
Exactly.
Yeah. All right. So one more time, in the name of our Inside the Hive.tv audience here, Lewis, thank you very much for your time. Thank you for the great work you're doing. And for you guys at home, thanks for watching Inside the Hive.tv, the show about bees. See you guys next week.